Hi there, welcome back to another video. This is going to be all about Mendelian genetics. And so we're going to go ahead and jump right in. This is just a review. Hopefully all of this already makes sense to you, but if not, buckle up. So let's go ahead and start over here on this corner. Now, depending on how much you know, Mendel was a scientist, and he was a scientist that did a lot of work with pea plants. And all of these studies really became the basis for what we understand is for genetics. Now, to be honest, we've built a lot upon this, and we've realized that there are some, or a lot, more subtleties to genetics. But this work is really important, and so that's why we cover it first. It's also the easiest part to understand, and then once we start putting in all these caveats, it becomes a little bit more difficult. And so, what did he start to come up with? Well, he came up with an idea of dominance. So dominance is the idea that one allele would make the organism exhibit the trait. In comparison to what you would need for a recessive, where you need two alleles. And this is typically represented with little letters, so little r, little l, little e, and any other letter that you can think of. And dominance would be represented with a capital letter. So I just need one capital letter, and I would exhibit the trait. Whereas here, I really need two. So I'm going to change this to say RR, little e. Okay, so this is the basis. And he was able to form a whole bunch of theories based off of this. He also came up with this idea of independent assortment. Independent assortment is the idea that chromosomes are going to separate independent of each other. This is really important because if chromosomes don't, and let's say all of the chromosomes that you originally received from the egg, aka the female parent, and the chromosomes that you receive from the sperm, aka the male parent, if they don't segregate independently of each other, then you are often going to get offspring that look just like mom or just like dad, and not a mix of the two, which is what we see for sexually reproducing organisms. And so this idea was really clever. This is a really good basis for all genetics to come. Now, let's go ahead and actually put it into practice. So, he did a lot of work in pea plants, and because these chromosomes segregate independently of each other, we can actually predict the offspring, the phenotypic ratio of the offspring for multiple genes simply by multiplying. So I'm going to briefly show you how the dihybrid works, but then I want you to remember that we typically are going to use probability rules rather than a dihybrid, because once you just get 3 traits, now you're already looking at a 64 square, and it just goes bigger and bigger and bigger and bigger, which is not what we want. So let's go ahead and look at this. So if I were to cross a round and yellow seed with a wrinkled and green seed, then these would be the individual crosses I would do. This is for round, this is for wrinkled, and this is for yellow, this is for green. When I do that, all of my offspring are heterozygous for both traits, which is exactly what we expect when we get the round and yellow. Now, how do I know it's round and yellow? Well, this idea of dominance. I have at least one capital letter here. Therefore, that's all I need to exhibit the trait. So this and this look the same, just like this and this look the same when we're actually looking at them. They have the same phenotype. Now, if I were to cross 2 round and yellows, but they're heterozygous, my cross is this. And the same is true for the yellow. I'm going to get a homozygous dominant, a heterozygous dominant, and a homozygous recessive for both traits. Now, that's exactly what we see here. Now, it's a little bit tricky because what they've shown in the dihybrid is the different gametes that you can make. And how do you get these gametes? Well, if you take your genotype, all you need to do is FOIL first, outer, inner, last, and you'll get these four for the gametes. And then same for the other side, these four for the gametes. Then when you combine them, you'll get big R, big R, big Y, big Y. So that is a yellow round seed. And you can do that for all of the squares here. But what I prefer that you do instead is do the probability rules. So if I want to figure out how many are yellow and round, so round, yellow, what I'm going to do is I'm going to figure out how many are round. Well, these 3 out of 4 are round. So that is equal to 3/4. Then I multiply by how many are yellow. Well, these 3 are yellow. So that makes 9/16 are the totally dominant phenotype. The yellow and round. I can do the same for all of the combinations I can make. And I do the round and green. Well, round we already figured out was 3 out of 4, but the green is actually 1 out of 4. So that is a 3 out of 16. The wrinkled yellow is, well, the wrinkled is 1 out of 4, but the yellow is 3 out of 4. So that's also 3 out of 16. And then wrinkled and green is 1 out of 4 times 1 out of 4, which is 1 out of 16. So this ratio, 9 to 3 to 3 to 1, is a very famous ratio that you should commit to memory. This is the ratio for crossing two totally heterozygous parents for 2 traits. In this case, it is seed texture, round versus wrinkled, and seed color, green versus yellow. So if you were to go and check the dihybrid, you'll find that 9 are round and yellow. You'll find that 3 are round and green, and then you'll find 3 that are wrinkled and yellow, and then 1 that is wrinkled and green. So you'll see that 9 to 3 to 3 to 1. It is, once again, worth committing that to memory because it will show up over and over and over and over again. So that, in general, is Mendelian genetics. You can assume independent assortment, meaning these genes are on different chromosomes and that these chromosomes are going to separate. And when they do that, they are going to follow simple probability rules like we see here. Now let's go ahead and do an FRQ that implements these ideas, and we're actually able to figure some things out if these ratios don't match what we expect with Mendelian genetics. What can we actually say about these genes? And so let's look at this one. This is from 2010, number 3. A new species of fly was discovered on an island in the South Pacific. Several different crosses were performed, each using 100 female flies and 100 males. The phenotypes of the parents and the resulting offspring were recorded. In cross 1, true breeding bronze-eyed males were crossed with true breeding red-eyed males. So let's go ahead and just dissect that first sentence right there. True breeding. This is another word for homozygous. Remember, homo meaning same. Same for this one. All of the F1 offspring had bronze eyes. Okay, so what does that tell me right there? So I have true breeding homozygous bronze-eyed males and true breeding females who were red-eyed. Now, if all the offspring have bronze eyes, that tells me that bronze eyes is dominant. Then we cross the F1 flies, and the data for the F2 flies were given in the table below. In case you don't know what F1 flies are, those are the offspring of the initial parents. The F2 flies are the offspring of the second generation, which is the F1s. So when we look at this table, they give us our F2 phenotype and the male and female breakdown. Now, oftentimes in AP, if they give you the male and female breakdown, that's a good sign that we might be dealing with something called sex-linked, which we're going to talk about more in our non-Mendelian lecture. However, there is a big trick with sex-linked traits. Sex-linked traits will show an uneven distribution based off of male or female. So in this case, if we look at bronze eyes, the males have 3,700 roughly. The males have 3,800 roughly. And for red eyes, the males have 1,260 and the females are 1,320. This is not, based off of my gut instinct, this is not a major difference between the two sexes. And so I would be inclined to think that this is an autosomal trait rather than a sex-linked trait, simply because I don't see that difference between male and female ratios. Okay, let's go ahead and look at cross 2. Cross 2 has true breeding normal winged flies. So now we're looking at a different traits. Before we were looking at eye color, now we're looking at wings. So normal winged males crossed with true breeding stunted winged females. All of the F1 offspring had stunted wings. The F1s were crossed, and the data for the resulting F2 flies are given in the table below. So looking at this, it's roughly the same again. So we have 1,160, 1,320 with normal wings, and then 3,600 and 3,800 with stunted wings. Okay, so in that case, that also is, that's also roughly the same. So I still think probably autosomal. Then we have true breeding bronze-eyed and stunted winged males were crossed with true breeding red-eyed normal winged females. All of the F1 offspring had bronze eyes and stunted wings. The F1 flies were crossed with the true breeding red-eyed normal winged flies. And the results were shown below. Okay, so we've got 4 phenotypes here, which is what we would expect. If we're dealing with 2 traits, you should see 4 phenotypes. So I'm going to figure out what the crosses are here. This is a true breeding bronze-eyed stunted male. So bronze-eyed was dominant. It looks like stunted wings were also dominant since all the F1s had it. So this is a totally dominant. I'm going to use the letters B, and I'm going to use the letters, let's do N. Oops. Okay, why did I pick these? I just like to pick letters that are going to be different for the capital and the lowercase version. So this is for bronze eyes. And they said it was true breeding, that's why it's homozygous. And this is for stunted wings. And we crossed it with a female who is normal winged and red-eyed. So they're going, the female, this is the male. The female is little b, little n. All the F1 offspring had bronze eyes and stunted wings. So all the F1s should have this genotype. This is the F1s. And if you're wondering how I got that, well, let's just do the 2 individual crosses. We have the big B from the male, the little b from the female. All my offspring are heterozygous for B. The same would be true for the big N's and the little n's. So these are all my F1s. Now these flies were crossed with true breeding red-eyed normal wings. So the F1, I'm going to draw the crosses over here. The F1s were big B, big N, little n. And then red eyes is recessive, and so is normal wings. Normal wings is also recessive. So it's like crossing back with the mom that's made here. So that'd be this cross. When I do that cross, these are my expected ratios. If you're wondering where these came from, the big B, little b came from the dad. The little b came from the mom. When I cross, I get this. So all my offspring, let's do the math here. So if they are going to be, I can get big B, big N, little n, big B, little n, little n, little b, big N, little n, little b, big, sorry, little n. These are my 4 options for genotypes here. And I just figured it out by saying, okay, I have these 2 options and these 2 options. Now let's go ahead and figure out the chances of each of these. So big B, big N, little n. It's a 50% or 1/2 chance for big B, little b. It is a 1/2 chance for big N, little n. So that is a 1/4. Same thing here, 1/2 times little n, 1/2. Maybe you're seeing the pattern that these are all going to be 1/2 times 1/2. So 1/4 should be each of these genotypes, aka 1/4 for each of the phenotypes. So this is bronze eyes, bronze and stunted. This is normal, wings, but with red eyes and normal. And these are 1 of each. The 2 in the middle are 1 of each. Okay. Now, these are my expected ratios. So I should expect 1/4 of my offspring to be each of these. Using my calculator, I'm going to add up how many offspring I have. Okay, so it does add up to 10,000. Here's why we don't really care now about the males or the females. Because we figured out each of these traits individually are likely to be autosomal, meaning they are not on the sex chromosome, not on the X or the Y, then I know that these should not still exhibit any sort of sex ratio here. I shouldn't see a difference between the sexes, and I still don't. And so I can actually combine these if I wanted to, if it makes the math a little easier. Okay, so let's go ahead and start answering the questions and see what else we need to figure out here. So what conclusions can be drawn from cross 1 and cross 2? Explain how the data supports your conclusions for each cross. So we've already answered this question. For cross 1 and cross 2, both of these traits, eye color and wing shape, are going to be autosomal. That's all I need to state for this first part, that these are autosomal traits. Now, how do I support that? Because I have an even number, a roughly even number between the 2 sexes, the males and the females. It's the same amount. If we saw a big difference, then I'd have decent evidence that maybe we have a sex-linked trait going on here. That's all I need for that one. Part B says, what conclusions can be drawn from cross 3? Now, cross 3 is a little different here. So cross 3, we expected a 1/4, 1/4, 1/4, 1/4 ratio here for the 4 different phenotypes. There's a big assumption that we make when we do these crosses with the probability rules. And maybe you recall at the beginning of the video what I said. We assume independent assortment, meaning that the genes are on different chromosomes. Now, if my ratios, my expected ratios, do not match what I observe, then I have evidence that maybe there isn't independent assortment. And we'll talk about in a second. The scenarios where that could be the case. Okay, so looking at the ratios, I don't see a match here. I see significantly more bronze eyes and stunted wings and red eyes and normal wings, and only a few that are bronze eye normal wings or red eye stunted wings. So since my ratio, my expected ratios do not match what is observed here, I have good evidence that independent assortment is not happening, which gives me good evidence that these genes are on the same chromosome. Independent assortment assumes they're on different chromosomes. Since this doesn't match my ratio, the genes are likely to be on the same chromosome. Now, that's not the only way to not see the expected ratios. We have other inheritance patterns that we might not have considered earlier. But in this case, I actually feel quite confident they're on the same chromosome, and here's why. If I see a significantly higher proportion of the parental phenotypes, what do I mean by parental phenotypes? Well, remember what these guys were. They were bronze and stunted wings, and this was red and normal wings. Bronze, stunted wings, red, and normal wings. These are the parental phenotypes. If I see a very high proportion of those, which is approaching 50%, then I have really good evidence they're on the same chromosome. Now, if they're on the same chromosome, they are not going to segregate independently. They are going to be together. And so if I see a high proportion of parental phenotypes, I am very confident that these two genes are on the same chromosome. So eye color, in this case, stunted wings / normal wings, are on the same chromosome. So how do I support that? My conclusion is that the two traits are coded for on the same chromosome. I support this by saying a couple of things. I have a couple of options here.
Okay, I want to jump in here and just let you know that we're not doing chi-square in this video, but you absolutely could use chi-square to support your claim here. I will do another video using chi-square with this problem soon.
I can say that the observed values do not match the expected values, which assume independent assortment. Therefore, they are likely on the same chromosome. I can, and I should, mention that we see a higher proportion of the parental phenotypes. And if you're really going for it, this is also considered evidence. You can mention how we get the non-parental phenotypes here. How do I get these mixed up ones? Well, maybe you recall, it comes back to this idea of crossing over. So these 4 sets here, the males and the females for these 2 different phenotypes, are the recombinant offspring. This is where if we had B and N on this chromosome and B and N on this chromosome, sometimes crossing over occurs and they do a little swap. So this chromosome takes this B and then they swap. And that is how you get recombination. We can actually figure out exactly how often these 2 traits are going to be, or 2 genes cross over, by taking these offspring numbers. Is 1,000. Oh, very nice. Nice round number. Okay, so this is 1,000. We had a total of 10,000 offspring. So we have a 10% crossover, 10% of the time. So maybe you remember that means that these 2 genes are 10 map units away between B and N. Okay. The last one here is a little bit beyond what we have discussed so far. We are going to talk about this a lot when we get to evolution, but for right now, that is where we will leave it. Hopefully that makes sense.